So imagine opening the hood of your car, and instead of the engine just sitting there, bolted to the chassis like normal, it has somehow hacked its own blueprint.
Right, like it's completely gone rogue.
Exactly. It literally sprouts its own autonomous wheels, detaches from the block, and just drives itself to another city to start building clones of itself.
Which sounds completely absurd. I mean, like a bad sci-fi movie.
It really does, but on a cellular level, that rogue, self-mobilizing engine perfectly describes the terrifying reality of how cancer metastasizes through the human body.
Welcome to our deep dive. Today we are tearing into a fascinating, cutting-edge 2026 oncology paper.
Yeah, published in the journal Cell, Death, and Disease by Zhang and colleagues.
Right, and our mission today is to unpack the precise molecular switch that gives lung cancer cells the structural ability to literally build legs and crawl.
It's a really striking way to visualize it, and unfortunately it represents one of the most stubborn hurdles in modern clinical oncology.
Yeah. To give you the specific context, this paper zeroes in on lung adenocarcinoma, or LUAD. Now, if you follow oncology, you know LUAD is the heavyweight. It's the most prevalent subtype of non-small cell lung cancer.
Making up roughly, 85% of those cases?
Exactly, 85%. And the frustration for clinicians is that despite having highly sophisticated targeted therapies at our disposal, like modern tyrosine kinase inhibitors, the 5-year survival rate for LUAD is still pretty grim.
Because the tumor spread, the primary reason for that failure inevitably points back to metastasis.
Spot on. If a tumor remained entirely localized, surgical resection or localized radiation would be highly curative.
Right.
But once those cells activate their motility programs and start migrating, the clinical landscape becomes exponentially more difficult to manage.
Okay, let's unpack this. We have to look at the mechanism of movement. How does a tumor cell, which is fundamentally just rogue biology, suddenly construct the structural apparatus to invade surrounding tissues?
Right, where does it get the tools?
Exactly. And Zhang's team hypothesized that the answer doesn't lie in mutating new genes, but rather in a microscopic, highly specific cellular tagging system.
That's the crux of their whole investigation. They focus on post-translational modifications, or PTMs.
PTMs. Okay, so if you're trying to picture this, think of a freshly synthesized protein as a capable but, totally blank worker.
I like that, a generic employee.
Yeah, and a post-translational modification acts like handing that worker a specific tool or slapping a sticky Post-it note on their back that changes their entire job description.
Right, it dynamically and reversibly alters what that protein can access, who can interact with, and its ultimate function. And the specific Post-it note this research focuses on is called SUMO.
SUMO, meaning small ubiquitin-like modifier. And the process of attaching the specific note SUMOylation, it relies on a very specific biological boss, an enzyme called UBC9.
Precisely. UBC9 is encoded by the UBE2I gene, and its role is paramount here. It is the only E2 conjugating enzyme in mammals capable of attaching that ISU tag.
Wait, the only one?
The only one. So if you bypass or eliminate UBC9, SUMOylation simply halts across the board.
Which explains exactly why Zhang and the team started their hunt there. When they pulled the bioinformatics data from the GEPIA database across multiple cancer types, they found that the messenger RNA for UBC9 is massively overexpressed in LUAD tissues compared to normal lung tissue.
It was a huge spike.
Yeah.
And that overexpression tracks directly with clinical outcomes. High levels of UBC9 strongly correlated with significantly worse overall survival and disease-free survival for these patients.
But they didn't stop at the databases, right? They moved to actual physical tissue samples.
Yeah, they needed to see what this overexpression was actually doing in real life.
Yeah.
So they analyzed 12 paired LUAD patient tissues, tumor tissue versus adjacent healthy tissue.
And when they ran the immunohistochemistry and Western blots on those tissues, the tumor side didn't just show a slight bump in UBC9. They saw a massive global increase in the entire pool of proteins carrying SUMO1 and SUMO23 tags.
The tumor cells were essentially hypersumoylated, just covered in these Post-it notes.
Which naturally leads to the interventional question, right? If UBC9 is the sole manager handing out these notes, what happens if we fire the manager?
Exactly. They utilized CRISPR-Cas9 to completely knock out the UB2I gene. They used two specific guide RNAs. SGUBC9 is 1 and 2, just to be sure.
And they did this in A549 and PC9 cell lines, right?
Yeah, both of which are highly aggressive LUA models that naturally express high levels of UBC9. They compare them to normal BCB cells.
And the functional collapse was absolute. When they ran CCK8 and colony formation assays, the proliferation of the knockout cells practically ground to a halt.
It was night and day.
But more importantly for our focus on metastasis, when they challenged these cells in transwell and matrixal invasion assays, the cells were effectively paralyzed. They couldn't crawl through the pores, they couldn't invade the tissue matrix.
And they corroborated this in vivo, too, in living animals. They took these UBC9 knockout cells, engineered them to express luciferase for live 3D imaging, and injected them into subcutaneous xenograft mouse models.
Oh, wow. So they could literally watch the tumors in real time.
Exactly. And the resulting tumors were severely stunted in both growth velocity and final weight compared to the wild-type controls.
So if I'm tracking the causality here, the LUAD cells are heavily overproducing this UBC9 enzyme. UBC9 aggressively hands out these SUMO post-it notes, and if we delete UBC9 entirely, the tumor loses its structural ability to move and grow.
That's the baseline, yes.
But I'm stuck on something. UBC9 is just the E2 conjugating enzyme. It's just the middleman applying the tags. Knocking it out stops the cancer, sure, but UBC9 isn't the actual engine driving the movement. If we're looking at a cellular network, who is it giving the note to that makes the cancer so aggressive?
That is the exact biological puzzle they had to solve next.
Okay, before we get to the prime suspect, let's do a quick recap so you can lock this in. First, LUAD is a deadly metastatic lung cancer. Second, UBC9 is a SUMO tagger that is massively overgrown in LUA. And third, disabling UBC9 stops tumor growth and spread entirely.
That's the perfect summary.
So, if you're listening right now, think about your own highly efficient workflows at your job. What happens if the person delegating the tasks goes into overdrive? How would you identify exactly who they are giving all that extra work to?
Well, the researchers deployed immunoprecipitation mass spectrometry, or IPMS, to do exactly that.
Okay, I always think of IPMS like dropping a highly specific molecular magnet into a cellular soup. You basically coat magnetic beads with an antibody that only grabs UBC9.
Right, it's highly selective.
Yeah, so when you pull that magnet out, you don't just extract UBC9, you drag out whoever UBC9 happens to be hugging at that exact moment. Then you run that catch through the mass spectrometer, which acts like a barcode scanner to identify the exact proteins you pulled from the wreckage.
I love that analogy. And when they scanned that catch, one prime suspect stood out from all the background noise. CORO1C or CORO1C?
CORO1C, which is incredibly telling because in cell biology, CORO1C is already a well-documented master regulator of actin dynamics.
Right. It's intimately involved in controlling the cell's structural skeleton. The physiological overlap is just too perfect to ignore.
So they had to confirm they actually interact, right?
Exactly. They immediately confirmed the physical interaction using a co-immunoprecipitation assay, or co-IP, in both the A549 and PC9 lines. UBC9 and CORO1C definitively bind to each other.
But physical binding isn't the same as functional modification. I mean, just because they touch doesn't mean the tag is being applied. They had to prove UBC9 was actually attaching the SUMO tag to CORO1C.
And to prove that, they set up a highly controlled cellular environment in HEK293 T cells. They introduced tagged versions of everything, his SUMO1, MyUBC9, and flag CORO1C.
And when they ran the Western blot analysis, the definitive proof emerged. A distinct protein band appeared that was exactly 15 kilodaltons heavier than standard CORO1C.
And 15 kilodaltons is the exact molecular weight of one single SUMO modifier.
Oh, there it is.
Yep. And to make the evidence totally bulletproof, they introduced the DSUMOylase called HA-SNP1 into the mix.
So SENP1 basically acts as a highly specific molecular eraser that cleaves SEON tags off proteins.
Exactly. And as soon as SENP1 was introduced, that 15 kilodalton heavier band vanished completely, confirming that CORO1C is absolutely a target of SUMOylation.
Okay, here's where it gets really interesting. Since we know the tag is being applied, the next logical step is figuring out exactly where it attaches, because the SUMO tags don't just stick anywhere on a protein, right?
No, they require a specific amino acid docking station, almost always a lysine, which is represented by the letter K in biochemistry.
So the researchers used a predictive database algorithm called JASA to scan the CORO1C structure, and they identified 3 high-probability lysine sites, K19, K311, and K440.
Right, and in biology, to validate those sites, you have to break them. So they systematically mutated these lysines, swapping them out for arginines, which are represented by an R.
Arginine is structurally similar enough that it doesn't fold or destroy the protein right, but it absolutely refuses to accept a SUMO tag.
Exactly, it blocks the docking station. But the biological redundancy they found here is fascinating. When they mutated just 1 or 2 of those lysines, say, they changed K19 to arginine to block the tag. SUMOylation still happened.
Wait, really? The 15 kilodalton band was still there?
Still there. The tagging system is incredibly flexible. If one docking station is structurally blocked, UBC9 simply pivots and utilizes a backup site.
That is ruthless efficiency. It shows how badly the cancer wants this pathway active.
It really does. So to completely erase the SUMO band, they had to engineer a triple mutant, altering K19, K311, and K440 all at once. This triple mutant is referred to as the 3KR mutant.
Okay, so with the 3KR mutant, they finally possessed a version of CORO1C that was functionally immune to being SUMOylated.
Yes, the tags could not stick.
So they reintroduced both the normal wild-type CORO1C and this new 3KR mutant back into the cancer cells. And interestingly, when they looked under the microscope using immunofluorescence, the location of the protein hadn't changed at all. Both the wild-type and the 3KR mutants stayed in the cytoplasm.
The spatial distribution was identical, yeah, but the functional capacity completely disintegrated.
The 3KR cells totally failed to promote proliferation, right?
Failed in proliferation, failed in xenograft tumor growth. But the most profound data came from the experimental intravenous lung metastasis mouse model.
Right, where they inject the cancer cells directly into the mouse's bloodstream, forcing the cells to survive circulation and actively invade the lung tissue.
It's a brutal test of a cancer cell's motility. The wild-type cells efficiently navigated the vascular system, breached the lung endothelium, and seeded aggressive, numerous metastatic nodules. The lungs were riddled with disease.
But in the mice injected with the 3KR mutant cells...
The lungs were almost pristine. Very few nodules.
That is wild.
It is.
Okay, time for another quick bullet recap. 1. UBC9 tags CORO1C. 2. It uses 3 specific lysine docking stations. 3. If you mutate all 3 into the 3KR mutant, the cancer loses its aggressive superpowers entirely.
Perfectly summarized. So if you're listening to this, I want you to think about this puzzle. If a protein is present in the exact same location in the cell, but it's just missing a tiny 15 kilodalton tag, how does it suddenly lose its ability to drive metastasis? What is that tag actually doing mechanically?
Right. To understand the mechanics of that failure, we have to look at the structural engine of cell motility itself, the actin cytoskeleton.
Which is my favorite part of this whole paper.
It's so elegant. So the best way to visualize actin filaments is to think of them like the continuous tracks on a tank, or tent poles being assembled. To crawl forward, a cell has to constantly build new tank treads at its leading edge, physically pushing the cell membrane outward.
And that actively crawling edge is called the lamellipodia.
But actin monomers don't just spontaneously organize themselves into the complex branching networks required for that push. They require a master architect to coordinate the build.
And in this context, that architect is the ARP23 complex. It binds to existing actin filaments and nucleates the growth of a new branch at a highly specific 70-degree angle. This continuous branching is what generates the dense pushing network required to invade.
And the upstream regulator that physically binds to and activates that ARP23 architect is none other than CORO1C.
Exactly.
So we have a very clean chain of command here. If you want a simple mnemonic to trigger your memory, just think of the letters UCA. U is for UBC9, which tags. C for CORO1C, which activates. A for ARP23. UCA, the cascade that builds the tank treads.
That's a highly effective way to map it, UCA. So the researchers needed to determine how the SUMO tag on CORO1C alters its interaction with that ARP23 architect.
And they did this with molecular docking simulations, right?
Yes. They found that the wild-type SUMOylated CORO1C binds to the ARP2 subunit with a very strong thermodynamic affinity, specifically negative 3.6 kilocalories per mole. But the untagged 3KR mutant, its binding affinity was severely compromised, dropping to just negative 1.7 kilocalories per mole. They proved this weaker physical binding with CoIP, too.
I have a question about this, though. So SUMOylation doesn't change where CORO1C is in the cell. We established that. It changes its shape, or like its stickiness, so it can grab the ARP23 architect. Is it really that simple?
It really is about structural stability. Without the tag, the 3KR mutant essentially slips off. It lacks the sustained binding required to keep ARP23 active.
And the visual evidence they provided for this is just stunning. They used immunofluorescence microscopy and analyzed it using Mander's colocalization coefficient.
Right, which basically measures how perfectly two proteins overlap in physical space.
Yeah. So they tagged CORO1C with a green fluorophore and ARP2 with a red one. In biology, when red and green perfectly overlap under the microscopes, they emit a bright yellow signal.
And what do they see in the wild-type cells?
In the wild-type cells, there was an intense concentrated band of yellow right at the very edges of the cell, the lamellipodia. They were perfectly locked together, exactly where the cell was actively building its actin tank treads.
It's beautiful to look at. But conversely, in the 3KR mutant cells, that colocalization just evaporated. The red and green signals were scattered diffusely throughout the cell, disconnected. The Manders coefficient dropped off a cliff.
And to see the tangible structural result of this failed connection, they utilized phalloidin staining, right? Which binds specifically to F-actin, the polymerized structural form of the protein.
Yes, in the wild-type cells, the phalloidin revealed these beautiful, robust, highly organized crawling edges. You could literally see the invasion machinery fully assemble.
But in the 3KR mutant cells, the actin cytoskeleton was just disassembled and messy. It was fragmented, completely disorganized, and incapable of generating directional force.
Which perfectly explains the paralysis.
Yeah.
If CORO1C cannot hold on to ARP23, the cell cannot build the actin network, and metastasis becomes physically impossible.
Okay, let's do our third recap. 1, CORO1C needs the SUMO tag to physically bind the ARP23 complex. 2, this binding builds the actin tank treads that allow the cell to invade.
Very concise.
So if you're listening, think about this. If we know exactly how the cell builds its engine for movement, how would you design a drug to throw a wrench in those specific gears?
Well, the Zhang team anticipated that exact question. They introduced a pharmacological inhibitor called CK666.
CK666?
Yeah, it's a cell-permeable drug, meaning it easily crosses the cell membrane. And its mechanism is highly specific. It wedges itself directly into the ARP23 complex, locking it in an inactive sheath.
It basically paralyzes the architect.
Exactly. So they set up a final, definitive experiment. They took the wildly aggressive wild-type cells and the paralyzed 3KR mutant cells and treated both cohorts with 100 micromolar of CK666.
And in the wild-type CORO1C cells, the inhibitor was absolutely devastating. It shattered the F-actin network, stopping colony formation, migration, and invasion dead in their tracks.
And they translated this to their animal models, too, utilizing intraperitoneal injections to deliver the CK666 systemically to the mice.
In both the localized subcutaneous models and the aggressive lung metastasis models, CK666 dramatically shrank tumor volume and virtually prevented all the metastatic lung nodules.
But here is the crucial detail, the true brilliance of this experiment. CK666 did absolutely nothing to the 3KR mutant cells.
Nothing at all.
No significant effect. Why? Because without a SUMOylation, the 3KR cells weren't using ARP23 to grow anyway. Their engine was already offline.
Wow.
If we connect this to the bigger picture, this proves unequivocally that the UBC9 and CORO1C axis relies exclusively on ARP23 hyperactivation to drive metastasis.
There is no backup engine.
Exactly. Its only lethal weapon is the ARP23 pathway.
So what does this all mean? Let's recap the entire UCA pathway from start to finish.
Let's do it.
LUAD overproduces UBC9. UBC9 then over-SUMOylates CORO1C at 3 specific lysines, K19, 311, and 440. That SUMO CORO1C then hyperactivates ARP23, which causes the cancer cell to build massive actin networks to crawl, invade, and metastasize.
It's a remarkably elegant molecular detective story. But, as with all high-level science, mapping one pathway immediately illuminates the blind spots in our understanding.
Well, for sure, there are still big unanswered questions here.
Yeah, for instance, the SUMOylation cascade usually requires an E3 ligase, a final helper enzyme that provides substrate specificity. This paper clearly demonstrates the role of the E2 enzyme, UBC9, but we still don't know which specific E3 ligase helps UBC9 this process in LUA.
That's a huge missing puzzle piece. And we're also left grappling with how this interacts with known phosphorylation, right? Because previous literature establishes that CORO1C is heavily regulated by an enzyme called CK2 at a site known as serine 463.
Exactly. So now we have a single protein governed by at least 2 distinct modifications, a SUMO tag at the lysines and a phosphate group at the serine. Deciphering that regulatory crosstalk, do they compete? Do they cooperate? Is undoubtedly the next frontier.
It really reframes how we view cancer progression. I mean, we spend so much time in oncology trying to delete or poison mutated genes that are broken beyond recognition. But here, the proteins are just doing their normal jobs, just with too many Post-it notes telling them to build.
Right. It's just bad administrative tagging.
Which leaves you with a really provocative thought to mull over. If cancer is sometimes just a disease of bad administrative tagging, what other deadly pathways in the body could be cured just by removing a microscopic sticky note?
It's a fascinating paradigm shift.
It really is. Well, thank you for taking this deep dive with us. Keep questioning, keep learning, and we will catch you next time.